Hello everyone, my name is Dr. Viktor Bauer. I'm a chief physician of the pediatric ward of the Dr. Kenese Albert Hospital in Hungary and one of the principal investigators of the 5 HMO study that I'm going to speak about right now. The study is about a randomized controlled trial testing term infant formula supplemented with a unique blend of 5 human milk oligosaccharides. I would like to start with some scientific background and rationale. Breast milk is a dynamic multifunctional biofluid containing not only nutrients, but also plenty of bioactive components that support health and development, of which human milk oligosaccharides is one of the most important. HMOs are non-digestible carbohydrates and represent the third largest solid component in the breast milk. More than 160 HMO structures have been classified already, and they have 3 main categories depending on the structure. First group is neutral, then the second group is fucosylated, containing a fucose molecule, and sialylated, containing sialic acid. Among the predominantly reported HMOs are 2-fucosylactose, or also known as 2-FL, 2,3-difucosylactose, or di-FL, lactoantetrose, or LNT, 3-sialylactose, otherwise known as 3-SL, and 6-sialylactose, 6-SL. Each HMO structure contributes to its function, resulting in diverse roles in supporting gut and immune health. There are emerging data that suggest that HMOs may also have a very important influence to the cognitive development. Therefore, is it of clinical importance to evaluate infant formula supplemented with a blend of the 5 HMOs, 2-FL, di-FL, LNT, 3-SL, and 6-SL. This HMO blend is designed to represent the major HMO classes and is based on the high HMO profile found in breast milk. The purpose of the study was to evaluate growth and tolerance in infants fed an infant formula with a blend of 5 HMOs. The results that I'm going to present now are for the primary endpoint, which was mean daily weight gain between baseline and age of 4 months, and the secondary tolerance endpoints. These secondary tolerance endpoints included stool consistency using a validated 4-point scale from a 3-day stool diary, gastrointestinal tolerance, gastrointestinal symptoms and associated behaviors from the 3-day gastrointestinal symptom and behavior diary, and also anthropometric Z-scores and the standard adverse event reporting. Okay, the study is a multi-center, randomized, controlled, double-blind study of healthy term infants conducted in 3 countries, Bulgaria, Poland, and in Hungary. Formula-fed infants were randomized into control group, test group 1 and test group 2 infant formulas, while breastfed infants were enrolled in parallel as a non-randomized reference group. Control infant formula was a standard cow's milk-based infant formula composed of intact protein and 100% of lactose. Test group 1 and test group 2 infant formulas were identical to the control formula, except that the part of the lactose was replaced with either 1.5 gram per liter of the 5-HMO blend in test group 1 and 2.5 gram per liter of HMO blend in test group 2. Formula-fed infants were exclusively fed formula according to the allocated treatment for 4 months, and the breastfed infants were exclusively breastfed for 4 months of age, until 4 months of age. Okay, this is the disposition of the participating infants in total. 626 out of 629 screened infants were enrolled, and 636 formula-fed infants were randomized, with 180 infants to the control group, 176 infants to the test group 1, and 180 infants to test group 2. 90 non-randomized breastfed infants were enrolled in parallel. The full analysis set included all randomized infants, except those who never get any feeding. So you can see in the table that there were 3 in the control group, 3 infants who never received the test formula. Therefore, the FAS is 177 infants. The per protocol set, or PP, includes all the FAS infants, but of course not those who dropped out during the study and not those who had major protocol deviations. Okay. On this slide, we have the baseline demographic characteristics, enrollment age, gestational age, mode of delivery, infant sex, maternal age, the years of the mother's education, which were all comparable in all feeding groups, in all formula groups. There were some minor differences in the breastfed group, which was slightly more vaginally delivered infants, and the mothers had slightly more years of education than the formula-fed infants. Okay, now I come to the primary endpoint. The objective of the study was to demonstrate non-inferior growth in terms of weight gain in gram per day in infants received in the formulas containing the HMOs compared to the infants who received the control formula from the enrollment to 4 months of age, according to the FDA guidelines in testing the infant formulas. To demonstrate this, the lower bound of the 95 confidence interval for the difference in growth between the groups and the control needs to be above -3 gram per day as defined by the FDA. As you can see in the table here, that was indeed the case both in the full analysis set and in the per protocol data set. You can see the numbers. It was above the non-inferiority margin of -3 gram per day, indicating that the formula of the 5 HMOs supports age-appropriate growth. Okay, z-scores. As seen on this figure here, age-appropriate growth was confirmed by the anthropometric z-scores, which were all comparable between the formula-fed infants and closely tracking by among the median of the WHO growth standards. There were no significant differences in weight for age, length for age, height circumference for age, and BMI for age z-scores between any of the formula groups throughout the study. Formula-fed infants had soft stooling pattern, as breastfed infants with the majority having predominantly loose stool throughout the study, and a very low percentage of watery and hard stools, as seen on this figure, which is for the 4 months of age time point. Yeah. Gastrointestinal tolerance and gastrointestinal related behaviors are shown in this table. The first graph describes a similar frequency of spitting up among all feeding groups. Then in the second table, you can see the incidence of gassiness. That was all comparable between the formula groups, except there was a slightly different in the breastfed group, which was slightly less frequent at certain time points when compared to the formula-fed infants control group. Crying and fasting behaviors were comparable in all feeding groups. Additionally, the mean total crying and fasting durations were very short and less than half an hour per day in all groups. And in the fourth graph, you can see the total time spent sleeping per day, which is comparable in the formula-fed infants and similar to the breastfed infants. Now, the adverse events, parents reported and physician confirmed overall adverse events were low and similar and comparable between the groups, indicating the safety of the HMO supplemented formula. There were no also significant differences in the incidence of serious adverse events in all groups, between the groups. And all serious adverse events were classified as unrelated or unlikely related to the study formula consumption. In conclusion, we can say that infant formula supplemented with a diverse blend of 5 human milk oligosaccharides supports age-appropriate growth and soft stooling pattern is safe and well tolerated. And with that, I would like to thank you for your attention.